Hi everyone, it's Mr. Sinti, and today I am happy to be discussing with you the process of cell respiration. Yeah, cell respiration is one of the most classic concepts in biology, and it's a biochemical pathway that is harvesting, which is kind of a cool word, it's harvesting energy from the food that we eat. And cell respiration obviously is occurring in the cell. And what's fascinating about it is some of the ingredients for cell respiration. Principally, we're talking about glucose metabolism in this video, is coming from the food that we eat, but ultimately it's going to get to the cell via the blood, and the oxygen is going to get to the cell via the blood as well. And so let's jump right into the conversation. So what's fascinating about cell respiration is that it's broken up into these classic three parts. They're sort of like movements in a symphony. You have glycolysis, and you have the Krebs cycle, and the electron transport. So this particular video is going to emphasize the pathway of glycolysis. And so these three metabolic stages are really interesting. And what's fascinating about this is that there's going to be some biochemistry involved, and I'm going to walk you through the 10 steps of glycolysis. And I'm going to refer to the structure of some of these molecules and even discuss some of the enzymes involved that catalyze it. But I think overall, the message that you should be getting is the big picture, the greater understanding, like just the profound importance of it. Because again, if you wanted to recall, things are different these days in terms of education. If you wanted to recall specific names of substrates or products or enzymes, they're readily available, like in a textbook or online. And so don't get too bogged down by detail, although I'm going to try to get to detail, but don't be bogged down by it. Sort of just take it in and enjoy the view. That's my opening remark when it comes to glycolysis. And so what I'm going to try to do now is animate, for example, what a typical cell would look like. If I were to draw the plasma membrane around like this, draw the plasma membrane. And so over here on the outside of the cell, of course, adjacent to every cell is the blood. And the blood's going to be, as I mentioned before, carrying glucose in it, and it's also going to be carrying oxygen in red blood cells. But let's just take a look at glucose for a moment. Now, glucose is going to diffuse into the cell from an area of high concentration to an area of low concentration. And so when glucose, let me just write it out, glucose right there. When glucose, which if you're familiar with this, is a 6-carbon hexose sugar, it's an aldose, monosaccharide. When glucose comes into the cell, it's going to undergo the first part of cell respiration, and that's called glycolysis. That's right. And the word glycolysis, if you were to break that down in terms of like what the meaning of the parts of the word, glyco means sweet, and lysis means to split. And so ultimately, pretty simply speaking, glucose is going to be cleaved or cut into 2 3-carbon molecules. And so it's a 10-step pathway, and it occurs in a linear fashion, and it occurs in the cytoplasm. So I'll write that down. It occurs in the cytoplasm, or the cell liquid, cytosol, and it occurs here. And each of the reactions is, of course, catalyzed, as we're going to see, by a specific enzyme. And so that's kind of neat too. So each of these, so it's substrate, product, substrate, product, substrate, product. And the end product is going to be, as I mentioned before, 2 3-carbon molecules known as pyruvic acid. And all of this glycolysis is the first phase of cell respiration, whether it be aerobic or anaerobic. Now, aerobic means with oxygen, and anaerobic means without. Glycolysis itself is part of aerobic cell respiration. Aerobic cell respiration is those 3 phases: the glycolysis, the Krebs cycle, and the electron transport system. However, in and of itself, it's an anaerobic pathway because there's no oxygen involved. I'll get into that in more detail in a separate video on the Krebs cycle and electron transport system. But if oxygen is available, if there is O2 in the cell, and how did it get there? From the blood. If there's plentiful amount of oxygen, then pyruvic acid will diffuse into the mitochondria. And inside the middle of the mitochondria, known as the matrix, is a cyclic chemical reaction, which is the second part of cell respiration, which is known as the Krebs cycle, which we'll get into that in a separate video. And then the third part happens in this inner membrane called the cristae, and that's the electron transport system or electron transport chain. And that's where a tremendous amount of energy is liberated from the food that we're eating, ultimately from glucose. And so, but this particular pathway that I'm going to emphasize is glycolysis today. And so let's get into that conversation. Let's get into that. And so as I was trying to describe and animate, glycolysis is occurring in the cytosol. And so though there's this 3 steps here, it's really a 10-step pathway that produces pyruvate or pyruvic acid. And I'll discuss the difference of that a little bit later. And so ultimately, this diagram is showing that ATP is being produced both in glycolysis and a little bit in the Krebs, but a lot of it is being produced in the electron transport system. And so I just don't emphasize that, but I just want to show you the locations. Glycolysis is happening cytoplasm, Krebs is happening in the mitochondrial matrix, and electron transport system chain is occurring in this inner membrane called the cristae. Right in there, and that's where the electron transport system is occurring. So let's get into this. So here's glucose. So glucose comes into the cell, and sort of like you enter the house and you're greeted. What's the first thing? Now, again, here's a picture of glucose. Let's get familiar with it. It's called 1, 2, 3, 4, 5, 6 carbons. It's a polyhydroxyl. It's an aldose sugar. Okay, so there it is, glucose. Comes into the house. It's greeted in step 1 by this enzyme right here called hexokinase. Now, hexo is referring to the fact that it's a 6-carbon, right there, 6-carbon sugar, glucose is. And a kinase is a classic, that's kind of a generic enzyme that's capable of transferring a phosphate. So it's capable of phosphorylation. So where is it getting the phosphate? So how do you like this? ATP, which is adenosine triphosphate, is present in the cell. And so what's going to happen is one of the phosphates is going to be transferred by the help of hexokinase from ATP actually to the substrate, which is glucose. And in so doing, when it sticks it on there, it's going to become glucose 6-phosphate. And that's in reference to the location of where the phosphate, as you can see here, functional group is attached. 1, 2, 3, 4, 5, and then 6. So it becomes, that's the product of the first step of glycolysis, glucose 6-phosphate. And of course, if ATP gives up a phosphate, it then falls down to adenosine diphosphate. So here's the thing. This first reaction is kind of maybe a surprise to you because it's using ATP, and you might have gone under the assumption, and you're right, that the main point of cell respiration is to generate ATP. It's to generate energy molecules from the food that we eat. But there is an energy investment. And so for the first 5 steps of glycolysis, we're going to see that there needs to be some ATP used. As a matter of fact, there's going to be 2 ATP used, just, spoiler alert. So 2 ATPs are going to be used, but then ultimately we're going to see in the latter half, the back 5, if you will, there's going to be a production of 4 ATP. So if you've used 2 and you make 4, you're actually going to net 2 ATP, which is not a terrific amount, but it's just the first phase of cell respiration. Just wanted to point, just a little shout out. One of the interesting side notes to the fact that a phosphate is added to glucose to make it glucose 6-phosphate is that, as you may know, the phosphate is negatively charged functional group, and in so doing, by attaching it there, it sort of restricts its movement, and so it's less likely to diffuse out of the cell. So it's kind of captured by putting a phosphate there. I find that to be kind of interesting. So over here on the left side, as you can see, it's kind of the big picture. So the first chemical reaction we saw, a phosphate added to glucose to make glucose 6-phosphate. And so over here on the right, it's showing like a close-up of what's happening in step 2. And so step 2, let's take a look at that. Step 2 is this molecule, glucose 6-phosphate, is greeted by phosphoglucoisomerase. There's a mouthful for an enzyme. So an isomerase enzyme is capable of transferring glucose into its isomer cousin, fructose, which is also a hexose monosaccharide. And so there's fructose. And so what do? The enzyme switches it around and it produces the 1, 2, 3, 4, 5, 6. So it's fructose 6-phosphate. And so what's the significance of this? Again, some of the steps, it's not a big one, but then again, it is. Meaning that not so much is happening in step 2 of glycolysis, but what's fascinating is if nothing much is happening, what it's doing is preparing it for what's going to happen next. And so this particular video is not on the level of a biochemistry class. So apologize for that without getting into too much more detail about it. But I'll just say that a step like this, converting glucose 6-phosphate to fructose 6-phosphate, is preparing for the next step. It's energetically more favorable in this particular sugar than it is in that sugar. Okay, so step 3. Step 3 is taking the fructose 6-phosphate. Here's fructose 6-phosphate, and it's going into the active site of this enzyme, phosphofructo, because it's fructose now, phosphofructokinase. Hey, another kinase. So you can anticipate another ATP is going to be involved. And so it gives up its phosphate and it becomes ADP. And so now you have fructose 1,6. Again, the 1 and the 6 is a discussion of where those phosphates are attached. So it's 1,6 bisphosphate. Now bis, just a little heads up on that. Sometimes, chemists will use the word bi, meaning 2. They'll use the word di, meaning 2, or use the word bis for meaning 2 as well. So it's 1,6 bisphosphate. In other words, 2 phosphates are over there. Now, phosphofructokinase, just a little shout out so you can see in step 3 here. Now we're in the whole 2 ATP. So if you wanted to keep track of that, you could say, okay, so we're minus 1 ATP, and then over here we're minus 1 ATP. So we're down 2 ATP. But if I can use this analogy, like if you're opening up a restaurant and your goal is to make some money by serving up food, you're obviously going to invest in the restaurant. You're going to decorations, tables, food, but then ultimately you'll make it back in the end. And so there's a little investment there of minus 2 ATP. But again, I was going to shout out for this phosphofructokinase. It's a rather large enzyme. It has a quaternary structure. Let me show you a picture of it. There's 4 subunits, and it's rather large. And what's interesting about it is it's a kinase, so it's capable of taking ATP and sticking it onto fructose. But it's also a regulatory enzyme, so it has the ability to be allosterically regulated. So it has alternative sites that when ATP is in a prolific amount, when there's a high abundance of ATP, it actually inhibits the enzyme's ability. So it becomes overwhelmed by the ATP, and when the ATP binds allosteric to it, it actually changes its conformation. And let me go back for a second. And so if it's not working, it's not going to then produce. Now, it's not going to take it out completely. It's not going to destroy it, but it's an example of feedback inhibition. When there's a lot of ATP, it's going to slow down its catalytic ability, and therefore fructose 1,6 bisphosphate will not be produced, and it's going to therefore shut down all of the other pathways that I'm about to discuss. So that's kind of cool that it's an allosteric regulator. And again, you'd want to put that early in the pathway in order to be economic about matters. Okay, so just to point that out. Okay, so now we're on step 4. Very exciting. So step 4 of glycolysis, and again, I apologize for being a little silly about some of these things, but step 4 to me is when fructose 1,6 bisphosphate enters an enzyme called aldolase. Okay, And aldolase will take the sugar and split it. So it's like a cleaver. It's going to cut the fructose. Let me just be dramatic. It's going to cut it. And so step 4 to me is sort of like the title track of glycolysis, because you remember I said that glycolysis literally meant like sugar splitting. And so it's splitting, although you're like, wait, glucose is being split. And it's like, yes and no, because the truth is glucose is way up here and it wasn't even involved. But ultimately, it was glucose that was being split, but really what's being split, technically speaking, is fructose 1,6-bisphosphate. So check it out. If you split it in two like that, this is a 6-carbon sugar. If you split it in two, what you're going to get is 2 3-carbon molecules. Perfectly good sense, right? 1, 2, 3, 1, 2, 3. And what are those products? Well, one of them is dihydroacetone phosphate, oxyacetone phosphate, DHP, the acronym for that, DHP. And then this guy over here is glyceraldehyde phosphate, sometimes known as GAP, glyceraldehyde phosphate. Or, apologize for this, sometimes depending on the text, it's known as phosphoglyceraldehyde or PGAL. I prefer GAP personally. Okay, so if you make one of these, you're going to make one of those. So those are equal amounts. And do you see how the phosphate, when you cut it, the phosphate goes over here, and then this phosphate goes over to here. So this is, let's just take a look at GAP for a second. Why is this? Again, we're not at this level of an investigation. We wouldn't be talking about this. You wouldn't be watching the video if you were scared. And so I bring no fear to biochemistry at this point, like bring it. Like glyceraldehyde phosphate, take a look at this. This is our friend, glycerol. We've seen this before. Glycerol is one of the main backbones to a triglyceride or a phospholipid. So check it out, glycerol. So it's usually a 3-carbon alcohol, but over here you could see it's an aldehyde, okay? At the end of the molecule, when it's a double bond O, this is what an aldehyde is, okay? So it's glyceraldehyde 3 because it's carbon 1, 2, 3. So glyceraldehyde 3-phosphate. All right. So check this out. What's step 5? If step 4 is splitting this up into 2, step 5 is an isomerase, just a general term. Isomerase, which converts DHP and GAP back and forth, back and forth, back and forth, back and forth. It can go back and forth. But if you recall your basic chemistry of Le Chatelier's principle, if the concentration of the product and reactants are the same, it's going to be in a state of equilibrium. And so the direction of these reactions are going to be back and forth equal. Okay? But what I'm about to tell you is rather important. The next enzyme that catalyzes step 6 of glycolysis, and I'll just draw it over here even though it's coming in the next slide. The enzyme that catalyzes step 6 is an enzyme called glyceraldehyde dehydrogenase, GAPDH. And so what's fascinating about that is enzymes are very specific, and if it has only I's for GAP, it's called glyceraldehyde phosphate dehydrogenase, GAPDH. If it only has I's for glyceraldehyde, that means it's going to be converting GAP into something else. So the concentration of GAP is going to be always low because it's always going to be used. It's always going to be going down. And so if it's low, that means that this direction of the reaction will be very minimal since it's not a lot of it, and this direction of step 5 would be major. So what I'm getting at is this. Though step 5 converts DHP and GAP back and forth, the truth is that for all intensive purposes, at the end of step 5, going into step 6, really what you have as a result of this, and I'm going to emphasize it, I'm going to go like red here, really what you have are 2 glyceraldehyde phosphates. You basically have two of these, okay? Because this is converted. I hope that makes perfectly good sense. Okay, so let's take a look at the very first 5 chemical reactions of glycolysis, because we're at the halfway point. So the first thing that happens, glucose comes in, ATP is used, there you go, glucose 6-phosphate, then it's converted to fructose 6-phosphate, and then hey, another kinase is involved, and so now you have fructose 1,6, split it in 2, and then hey, you actually have 2 glyceraldehyde phosphates. And so now we're going on to step 6. Yeah, not too bad, right? So step 6 is a biggie. You're like, well, nothing of major significance has happened other than you've invested 2 ATP. Well, step 6 is a pretty cool one, and let's get into it. Well, this enzyme right here that I was alluding before is called glyceraldehyde phosphate dehydrogenase, although sometimes it's generically called triose because it's a 3-carbon sugar, triose phosphate dehydrogenase. I just want to put a shout out is that this particular enzyme is rather important to me and in the school that I teach, because we actually have a class studying redwood tree genetics, and we're actually looking at the gene sequence of coastal redwood trees for this particular enzyme, so GAPDH. And so we're trying to compare genetic diversity between redwood trees by looking at that particular enzyme. So it's a personal favorite of mine. Just wanted to let you know. So what's going on with this GAPDH? GAPDH. So what is it doing? Well, first off, the substrate is glyceraldehyde phosphate, and what it's doing is it's catalyzing our very first redox reaction. Okay, and so a redox reaction is oxidation reduction. So who's losing electrons? Who's Leo is losing electrons? So who's losing electrons is going to be glyceraldehyde phosphate. And in so doing, and losing the electrons, who's gaining them? There's a coenzyme sitting in the active site of GAPDH, and it's called nicotinamide adenine dinucleotide, abbreviated NAD+. Notice how there's 2 there, because there's 2 substrates. There's going to be 2 from everything going on at this point. So what happens is a redox reaction, so it's losing its electrons, and who's gaining them? NAD+ is gaining them, and in so doing, gaining electrons means that NADH is going to be reduced. So you make 2 NADHs, and of course 2 protons, because it's the hydride that's actually being transferred, and I'll write that in. So hydride is coming over, and that's why you're getting this. Now, NADH is a storage of energy, because the electrons are being held more closely than they were in glyceraldehyde phosphate, and so some of the energy is now being transferred to NADH. Here's a little shout out for what's going to happen later in a separate video. The NADH that is produced is then going to be used in the mitochondria to produce a tremendous amount of ATP. Okay, but in and of itself, it seems insignificant that NADH is being produced. But I'm just saying that will be used later to make a lot of ATP. So a redox reaction often releases energy. Sure, it's being stored in NADH, but it also releases other energy. And the energy that it's being released is then utilized by also the enzyme, GAPDH, by taking an inorganic phosphate. Do you see that inorganic phosphate? That means that it's not connected to any carbons. So it grabs, and there's 2 of them because there's 2 GAPDHs. It grabs it, and although there's only 1 shown here, it's going to stick a phosphate onto it. So do you notice how GAP has only 1 phosphate on it? So it's going to stick another one on it, and so now there's going to be 2 phosphates. So it utilized the energy of the redox to take advantage and now put 2 phosphates onto the product. And what's the product of this redox reaction? The product is, there's 2 of them, 1,3-bis phosphoglycerate. So it's a glycerate at this point. Okay, so that's the end product of step 6, which was a redox reaction. Okay, so here's the 1,3-bisphosphate glycerate. Okay, so step 7 is a kinase, but check this out. It's phosphoglycerol kinase. This particular time, we're now going to rip a phosphate from the substrate and stick it onto ADP to produce ATP. So we're going to actually transfer from the substrate. Finally, we're going to use the substrate's phosphate to make ATP. Now, this is an important moment. This type of production of ATP is appropriately and maybe obvious called substrate level phosphorylation. Substrate level phosphorylation. So in other words, you're using ATP. It's the most primitive way to make ATP. There's going to be another way that we discuss in the electron transport chain called the chemiosmotic production of ATP, or oxidative phosphorylation. But this is called substrate level. So you're just transferring it over, and the kinase is doing it. So you make 2 ATP, and you're like, why are you making 2 ATP? Because there's 2 of these things. Okay? So 1 and then 2. That guy still stays there. And again, there's 2 of them. So now the end product here is 3-phosphoglycerate. And so if you're keeping track of your ATP, you used 2 and you made 2. Now we're at 0. Step 7, and we haven't even done anything in terms of making ATP. So then I mentioned, like, sometimes the reaction doesn't seem like much is going on, but see, check this out. Step 8 goes 3-phosphoglycerate goes into phosphoglycerol mutase. How's that for a name? And what it's doing is, if you take a look here, and without me even describing it, what do you think is happening? Like when you take a look at the phosphate is on the 3 carbon, and then look over here, the phosphate's now on the 2. You're like, yeah, it doesn't seem significant. It doesn't seem too much to me either, but a biochemist would see it a little differently. I suspect that in the movement of the phosphate over to the 2nd carbon, it's going to make step 9 reaction more energetically favorable. That's a bit of a cop-out answer, but it's true, I think. So step 9, enolase, is going to take the 2-phosphoglycerate, and what it's going to do is it's going to remove water. Okay, so how about that? A little water is produced in glycolysis. Handy-ho. But it produces 2 waters. Actually, it only produces 2 because 1 per molecule, because there's 2 of everything, so a water is removed. And what's the significance of the water being removed? The significance is that you're losing that hydroxyl group, and you're putting a double bond over here. And again, you're sort of priming what's going to happen in the final step of glycolysis, which is step 10. So this product right here is 2-phosphonyl pyruvate. It's sometimes referred to as PEP, and it's fairly famous. It's going to come up a couple of more times in our discussion of biochemical pathways in separate videos. So PEP has a phosphate on it, and check this out. What happens to PEP is that it takes a kinase. It goes into the active site of a kinase, and curiously, this is called pyruvate kinase. I would have called it, I don't know, necessarily PEP kinase, but it's called pyruvate kinase. And then what it's doing is it's grabbing that final phosphate off of PEP, PEP, and it's removing it, and it's attaching it onto ADP to produce 2 ATP. So that means we've now made 2 ATP, and this is also called substrate level phosphorylation. Substrate level because PEP is the substrate, and we're transferring it onto ATP. And here it is. The final product of glycolysis is pyruvate. Right there. It's a 3 carbon. Remember, there's 2 of them. We haven't lost any carbon in this pathway. We started off with 6 glucose. Now we have 2 pyruvate. Okay? And as you can see, it has a negative charge on it. And so just a little summary of this. And so here's the first 5 steps. So ATP is used, another ATP is used. We split fructose bisphosphate into 2. It's GAP, which is over here, that goes on. And then GAP, again, review, is being a redox reaction. and then some ATP is produced, 2, so it's net 0, and then 2 more are produced. So we have a net production of 2 ATP, and the final product is pyruvate. And do you notice we didn't use any oxygen? No oxygen at all in glycolysis, though it's part of aerobic cell respiration. It's the first part of aerobic cell respiration. But it's also the first part of a fermentation reaction, which I'll get into in a separate video. But glycolysis in and of itself is an anaerobic pathway. There's no oxygen. And it's an ancient pathway. And when I say ancient, I mean that it evolved very, very, very early on because it's very ubiquitous, meaning that all living organisms, every living organism on the earth has glycolysis. It's pretty awesome that way. And so here's a picture of pyruvic acid. And so I want to, what's the difference between pyruvate and pyruvic acid? So first off, just to refresh you, this little point right there is a carbon. So there's a carbon there, and a carbon there, and a carbon there. What a surprise. 3 carbons. And there's 2 of these, which are the end product. Now, over here, I put a little model over here to give you a little 3-dimension. But do you see how there's not shown in the picture 3 hydrogens coming off? It's understood. 1, 2, 3 over there. And then here's this central carbon with the double bond oxygen coming off right there. And then do you see, let me animate it. Come up with blue. Do you see this carbon right there? Okay. That one, when you have a C double bond oxygen OH coming off of it, that's a carboxylic acid. Do you see that? The tendency of a carboxylic acid sometimes is to have a proton jump ship, and as a result of it, what you get is COO minus. And in that state, in that anion state, it's referred to as pyruvate. But when it's like this, it's an acid. So it's pyruvic acid when it has the hydrogen, but then when it loses the hydrogen, it's considered to be pyruvate. Okay? Just wanted to point that out. Okay? So that's kind of significant. And so here's the second half of glycolysis where you make 2 ATP, so it nets 0, and then you make 2 more, and so you get 2 ATP, you get 2 waters, and 2 pyruvates. And just to emphasize this, the way in which ATP is produced is called substrate level phosphorylation. In other words, here's an example of that. We saw this. There's an enzyme, a kinase, that's capable of taking a substrate, in this case it's PEP, and ripping the phosphate and sticking it onto ADP to produce adenosine triphosphate in the product. This is the last step of glycolysis. You may remember it since it's fresh. This is step 10, and the production is pyruvate. And again, it's pyruvate because, as you can see, it's C, oxygen, double bond oxygen, like that, and the CH3. So it's pyruvate. If you put a hydrogen there, it would be pyruvic acid. Okay? And so I like this picture because it kind of summarizes things. So the first 5 steps, it's an energy investment phase. You use 2 ATP, and then ultimately you make 4 ATP, but really you net 2. And then you also make 2 NADHs, and again, that's significant. The NADHs will be cashed in later in the mitochondria to produce even more ATP. But the truth is, and you also make 2 waters. The truth is, there's a lot of energy still left in the pyruvic acid. And as it turns out, not only is the pyruvic acid going to go into the mitochondria if oxygen is present, that's going to produce a lot of ATP, and the NADH is going to help produce a little bit more ATP as well. Okay, so glycolysis, as a summary in conclusion, is a conversion of glucose to pyruvate, and it oxidizes it. Although, technically, oxygen isn't involved, and technically glucose isn't really what's being oxidized. If you remember, it was glyceraldehyde phosphate that was oxidized. But during glycolysis, in general, glucose is split into 2 3-carbon sugars. That's true. And those sugars are then oxidized and rearranged to make 2 molecules of pyruvate. So this is a summary. It seems kind of simple after going through it step by step. There's 10 steps in glycolysis. Each are catalyzed by a specific enzyme. It occurs in the cytoplasm. It's an ancient pathway. It's ubiquitous throughout all of life. It's pretty strong evidence for a common ancestor, and that all life is related. I wish Charles Darwin was able to see this video on glycolysis. He would enjoy it. I knew it. And so there's two phases, energy investment and energy payoff. And so there's no CO2 produced. And again, I think as a beginning student, you're like, wait a minute, I thought cell respiration was glucose plus oxygen makes carbon dioxide and water. It does, but there's no carbon dioxide being produced in glycolysis. It will be produced in the mitochondria in a separate video. Okay? It will be. And the truth is, as you saw, glucose doesn't react with oxygen. Okay? And so that's kind of a misnomer too. There's no CO2 produced, and glycolysis occurs whether or not there's oxygen present or not, because it's an anaerobic pathway. But if oxygen is present, then pyruvate acid would then go into the mitochondria when oxygen is present. So with oxygen, pyruvate acid goes in. Without oxygen, pyruvate acid stays in the cytoplasm, and it's either converted to, here's a little foreshadow, alcohol if you're yeast or bacteria, or lactic acid if you're an animal. Okay? Or actually some bacteria as well can do that. And so those will be separate videos where we look at the Krebs cycle, which occurs in the matrix, and the electron transport chain, which occurs in the crystal. Those are really cool. So in conclusion, I want to maybe make things a little bit more clear by showing you a little animation. And so let me do that. And so check this out. So you start off with glucose, and let me just do this. So you start off with glucose, and in the cytoplasm, there's a little ATP invested. So there's one phosphate attaching onto it, glucose 6-phosphate. And then it's not going to show a lot of detail, but then that fructose is broken up into two molecules. Basically, gap is what we're talking about there. There's two of them. And so then there's going to be an investment of a redox, which we're going to make two NADHs. That's kind of neat. Well, again, in the cytoplasm. Then we're going to make two ATP, so we've net 0. Do you see up there? We have a net of two, and then we're going to rearrange the phosphates onto the second carbon by removing water, and then the final step of substrate level phosphorylation, we produce 2 ATP. So in the end, we've produced 2 ATP, we haven't used any oxygen, and we've produced 2 NADHs. Pretty cool. And then one last one to finish, just to drive the point totally home. Here's a little look at glycolysis here. And so there's our sugar and our oxygen in the blood, and it travels in. It's a muscle cell, travels into the cytoplasm. Notice it's not going into the mitochondria right there. And what's going to happen in glucose? It's not going to react with oxygen, but a phosphate is going to be stuck on it. And again, it's not showing detail. It just showed a couple of steps all at once. So basically, you got fructose 1,6 bisphosphate right there, and then you're going to split it into 2. Okay? And so there you go. You split it into 2, although for clarity, the video is going to just show 1, but there's 2 of everything. Okay? So that little shiny yellow is the phosphate. And then how about that? There's NAD+. And again, NAD+, don't be worried about it. NAD+ is a very electronegative coenzyme, which then grabs electrons. Now it's reduced, so it's NADH. Okay, and then another ATP was used. How about that? Okay, so now you're going to remove the phosphates. And so what's going to happen here is the last phase of glycolysis, the energy production phase, is then you're going to make an ATP and then make another ATP. And so you're like, whoa, gee, only 2. Well, they didn't show the other one. So there's a total of 4. So you net 2. And then this is our good buddy pyruvate. And then if oxygen is present, pyruvate's going to go into the mitochondria. That's right. And inside the mitochondria, it's going to go into the Krebs cycle. So I hope you enjoyed this conversation about one of the great pathways. I hope I was able to do it justice. I feel extra pressure when I'm discussing something as important, something as profound, and just so critical, glycolysis. And so I hope you learned something, and more importantly, I hope you enjoyed it. Thanks for watching.